Hello, this is lesson 2 for Biology 3 Exploring Diversity. Our lesson is on comparing systems of classification. So, for example, in the picture shown below, a turtle can be closely related to fish on the premise that they are able to swim across the water column, so they are nectonic. However, if we try to consider another feature, say for example, on the basis that they lay eggs, at that they have large scales, and other quintessential characteristics of reptiles, then they can be more closely related to lizards, geckos, and so on. For this lesson, we should be able to differentiate the three types of classification schemes: phynetic, phylogenetic, and cladistic systems, and be able to compare and contrast the resulting trees, which are called phenograms, cladograms, and phylograms. In the previous lesson, you were taught about the basics of phylogenetics, which is simply establishing relationship between and or among organisms by considering the shared characteristics. So, one of the many early forms of classification of organisms are the ones projected on the screen. We have here in the left, which is a plate from Systema Naturae by Carolus Linnaeus, wherein he classified insects based on their wing pattern and then in their wing morphology. So some of the insects here have a single pair of double pair of wing as shown on the pointer, single, some have short, some have narrow, some even have printed patterns in it. While on the right, this is a famous example of the Galapagos Islands finches by Sir Charles Darwin, wherein he was able to observe differences in the beak size and girth. So these are the earlier forms of classification, and classifying organisms make them easier to study, which is why the study of phylogenetics and systematics are important for higher learning. So in these examples, there are two terms that you have to look into: character and character states. When we say character, it's usually a property, one of the many properties of an organism. While for character state, it's an interpretation of the feature or the observable part of an organism, which is used to discriminate the characters between organisms. So let's take the example of the finches. The character for this will be the beak. Okay, the type of the beak that the finches possess. The character states can either be pointed, can either be short, can either be wide, can either be, let's say this is narrow. Okay, so there are currently two systems because phylogenetics is further divided into two. So the earliest of which is numerical taxonomy, also called phonetics, and the classification is based on phenotypic similarities without reflecting evolutionary history. So it's simply taking into account how many characteristics are similar, how many characteristics are not. If we are only considering very few characters, say for example 1 or 2, then that is an artificial type of phonetic classification. So for example, in the picture, if we are only considering the number of appendages of an organism and then we are going to relate it to another organism, so that is an example of artificial. However, when we say it's a natural phonetic classification, our classifications are based on morphological and anatomical characters that are many at the same time. So if we try to compare artificial and natural, natural classification gives a better resolution of the organisms that we are to classify, simply because we are considering many character states at once. Now, phylogenetic classification is also called phyletic classification because if we try to compare it with phonetic, this tries to reconstruct evolutionary relatedness apart from shared morphological and molecular similarities in data, which can be further divided into phyletics and cladistics. So say for example, in my experiment on the COX-1 gene that is shared by tuna species mitochondrial genome, we can be able to make a phylogenetic tree by simply looking at the nucleotide sequences. So for example, in here, the first base pair is one character. Okay? So we can see here it's all G for the 23 tuna species and so on. Now for the third character, some are A, some are G, and some are D. So these are what we call character states. So if there are differences in the character states, then we can resolve which is closer and which are not. Okay? So this is just an example for this. For cladistics, so di ba under phylogenetics there are two, cladistics and then yung ating phyletics. Yung cladistics naman, it shows evolutionary history. However, the branch length does not matter. Okay? So the species that are placed in groups are what we call clades. So the ones in the colored rectangles are considered to be one clade. So this can be one, this can be one, and so on and so forth. Okay? Depending on which characters are we looking into. Say for example, in the basis of hair and fur. So rodents and primates are considered in one clade on the basis of the presence of amniotic egg. So reptiles, birds, rodents, and primates can be considered in one clade. So closely related organisms are represented by shared features that are called the shared derived characters or traits. So ito yung mga naka-highlight in pink. Next, phyletics. So yung phyletics naman, may additional feature lang siya. Other than establishing evolutionary relationship, we're also taking into account the length of the branches. So, madali lang tingnan which is a phyletic and cladistic classification kasi phyletic classifications later on, meron siyang value sa nodes, meron siyang scale sa branches. Okay? So there are many trees used in depicting relationships. So depending on what type of classification are we using, we also generate different trees. Okay? So for example, for phonetic classification, we get a phenogram, which is basically clustering of taxa based on phenotypic similarities. So for example, we have the character table, which was used to calculate the similarity coefficient K, summarized per species in table 2, which is the distance matrix. So for K is simply the number of similar characters between the two taxa. So kung titingnan natin yung ating pag-resolve ng similarity is by considering a pair at once. So for example, to solve for K between X and Y, we simply look into the number of characters that are similar for X and Y. So for example, for character state 3, so since 0, 0 sila, ibig sabihin they are similar for this character. Okay? Yung character trait nila for character 3 are the same. Okay? That goes the same with 4 and 5. That's why if we are going to compute the distance matrix between species X and Y, 0.60 yung lalabas. Simply saying that for the 5 characters considered in comparing species X and Y, 60% similar sila. So, ganito lang yung computation nito. And once we have the phenogram, we can actually make a cladogram by um, looking into the distance matrix. Okay? So, next is cladogram. So, balik muna tayo sa phenogram, ano? So, this is an example of how a phenogram can be made based on the distance matrix. So we can show here 60% similarity of traits between X and Y. That is why if we are going to plot it, nasa 0.6 siya. So all you need to do is to make an x-axis and then plot your x, y, and z. So between X and Y, they share 60% similarity as shown here. Kaya dito sila. Okay? How about for X? So for Y versus Z. Y versus Z, isa lang yung character trait nila na similar. That is why 0.2 lang sila. Okay? Between Y and Z, also the same point 2. That is why nasa point 2 lang din sila. So this can shift depending on the distance matrix that you use. So you can do this as an exercise if you're going to consider two or three similar characters traits, then mag-change yung ating distance matrix, and then mag-change din consequently yung ating phenogram. Next is cladogram. Sinabi natin kanina pag cladistic classification, pinapakita lang nito yung hypothetical relationship between taxa. So hypothetical because a phylogenetic tree, specifically a cladistic or phyletic classification, is only true on the premises of the organisms and the characters considered. So beyond this, hindi na nagiging totoo. Kapag nagdagdag ka na, so nag-shift yung ating phylogenetic tree, which will be different from the original one. So this type of classification also assumes the least number of character changes which have occurred when explaining a dataset. Ito yung tinatawag natin na principle of maximum parsimony. Yung naalala ko dito is yung explanation na if you cannot if you can't explain a concept in simple terms, then you simply do not understand it. Ibig sabihin, yung simplest way daw of evolution is most probably the correct way, which is why parsimony took took takes into account the minimum number of assumptions for the species to to evolve. So later on, we are going to have many examples for this one. So assumptions of cladogram, it's any group of organisms that are related by descent from a common ancestor. In this case, yung ancestor can be here, no, rooted. So all other organisms here are hypothesized to have originated from this common ancestor. And you can also see that there is a bifurcating pattern of cladogenesis. Like for example, here D and E, hindi siya pwedeng magtatlo sila because if magtatatlo sila, that should be further resolved. So more characteristics have to be looked into in order to resolve clade na meron siyang tatlong organisms. And the changes in characteristics are assumed to have occurred in lineages over time. So an example of cladogram here is the one that I made when comparing a gene of various organisms. So ito naman siya, I simply plugged in the molecular data. So basically, raw sequences, and then I did multiple sequence alignment, and then basing on the sequence, I was able to come up with a tree that differentiated vertebrate and invertebrate counterparts of this gene. The gene that I'm studying is actually an immune-related gene, which is pentraxin. So just by looking at the molecular data, I can be able to say here that the pentraxin gene of invertebrates were able to evolve separately with their vertebrate counterparts. So, ganun lang yung pinapakita ng cladogram. However, yung kulang sa cladogram is wala siyang time component. Okay? And this is where a phylogram comes in place. So, similar siya with cladogram pero yung branch lengths niya are proportional to evolutionary time. So again, I'm using molecular data kasi ito yung usually ginagamit ko. So this is also an example of molecular data. In here, it's actually a genome, a whole genome of Nemavirus, which is a pathogen for marine organisms, especially pinnate organisms. Okay? So nakikita nyo, yung pinagkaiba niya sa cladogram, merong scores yung ating nodes. And this is simply sequence similarity. Okay? Ibig sabihin, at this point, 100% similar ang Pineus monodon na Nemavirus with that of its um, relative, the Phaneropineus mergensis, and so on. Ganun lang yung sinasabi dito. Ngayon, makikita natin yung point 4. Point 4 is simply the scale of the similarity. So, at this specific branch length, sinasabi lang nito na it's point 4 similar in terms of the nucleotide or protein sequences. Okay? It's just how you interpret a phylogenetic tree. So as a summary, we have three major systems used to classify species. We were able to talk about phenetic, cladistic, and phylogenetic types of classification. So cladistic and phyletic systems are grouped under phylogenetics since parehong systems assume evolutionary relationships of the taxa being studied. And depending on the type of system that we are using, we are able to generate different trees. So phenogram, phenotypic relationship lang siya. Cladogram, meron siyang evolutionary relationship na na-establish based on shared derived characteristics. But for phylograms, kinoconsider nito both ang common ancestry as well as evolutionary time. Okay? Now, for us to learn more about this topic, you can watch the following videos at your most convenient time para naman meron tayong external input for this lesson. So, thank you very much.